Hey, hello everybody, and welcome to the Mitosis Notes Podcast, Lecture 1. I'd ask that you take notes like you do in class, and the answers will appear in red on the screen. Our story begins with the cells that we've taken a look at in class already, such as these onion cells here. We've also taken a look at skin cells from your wrist, and also bacteria cells in yogurt. Now, all of these cells we needed to use a microscope to see because they're so tiny. Now there is a cell that you can see with the naked eye, and this is from the chicken or egg cell, or chicken or ostrich egg. Now what you're looking at here, circled in red, is the chicken's egg cell. It's not the entire egg, it's just that little white dot. But aside from this, almost every other cell that you need to look at, you need a microscope to see them. So what's up with that? Cells tend to be really tiny because if they were too big, it would take too long for nutrients and wastes to get into and out of the cell. If it takes too long for organelles to get their nutrients, they would die, and the cell wouldn't be able to support itself. So if that happens, the cell is going to go through a process called mitosis to divide that big cell into two smaller cells so that the cells can survive. And now there's some other reasons why you would want to do cell division other than keeping the cells small. The first of these reasons is for growth. So we have a picture of baby SpongeBob and Patrick here. Now these guys weren't always this size. They had to grow up into adult SpongeBob and Patrick, and that means you would physically need more cells for that to happen. The second reason is to replace older damaged cells. So this adorable little kitty has a cast on, he broke his leg, and what's happening inside of his leg is his bone is making new bone cells to replace the damaged ones and to repair his bone. Now, not all cells will divide at the same rate. So, for example, these skin cells here in the upper left-hand corner go through a lot of wear and tear, so they divide every 1 to 3 days to replace the old ones. Something like your brain cell on the bottom here, you need more long-term, so these are going to rarely divide. If they do divide, it's very slow. So not all cells will divide at the same rate, and generally they make the same types of cells. So skin cells make skin cells, brain cells make brain cells. There are some cells in your body, however, that are able to make a wide variety of cells when they go through the cell division, and these cells are called stem cells. These would be the same cells that would be in your flatworms that allow them to rebuild their head and tail. Now stem cells are classified on what type of cells they can produce. The first of these types is a stem cell that can produce an entire organism from one cell, and these are called totipotent. These would be the cells in your worms. The second would be a cell that can produce any type of cell in the body, and these are called pluripotent. And the last type would be a stem cell that can produce only a limited range of cells, and these are called multipotent. Now in humans we have two sources of stem cells, the first of which is from embryos. Now embryonic stem cells are totipotent or pluripotent, so they can make all of the cells in the body, which makes them extremely useful. But because you have to take it from the embryo, it would kill the embryo and result in the organism's death. So because of this, the Church does not support embryonic stem cells. The other source is adult stem cells, and these are going to come from the bone marrow of individuals or from umbilical cord blood from babies. Now these particular stem cells are multipotent, so they don't make as many types of cells, but as you can see within the picture, it's a pretty good range. So these are very useful. They don't harm any embryos in the process, so the Church does support these. Now before the cell goes through cell division, it's in a state called interphase. This is the part of the cell's life when it's not dividing, and it's composed of 3 subphases. The first of these is G1. This is the longest part. The cell's doing normal cell functions like making proteins and growing. So this is basically the cell's life. The second part of interphase is the S or synthesis phase, where DNA is copied in preparation for cell division. We need 2 sets of DNA for each of the cells. And the last part is G2, where we make organelles for each cell that we're going to create in cell division. Now DNA is stored in interphase as chromatin, which is just a loose mass of the DNA strands. If you take a look at the cell above, in the nucleus, it just looks kind of fuzzy. Now when the cell is ready to divide, it makes the copies of DNA in the S phase, and it makes organelles in G2. Right now, all of that chromatin in the strands is really hard to move around, so we're going to tightly wind it into structures called chromosomes, so it's a little bit easier to move the DNA to each of the cells. You can think of chromatin as a mass of DNA strands, like this yarn here, and then chromosomes would be that chromatin tightly wound into these skeins of yarn, these tightly wound structures. Now, when we visualize cells under a microscope, we stain them to see them. And when that happens, these bands appear on the chromosomes, which are basically the genes being visualized. Each of the bands is a gene. So we have a gene here, and a gene here, and a gene here. Each of those are genes. If you remember, they're just segments of DNA that control a particular trait. Right, now in interphase we copied the DNA, so that means when we make chromosomes we end up with an original chromosome and a copy of that chromosome. And now usually together, when you have the original and the copy, these are called sister chromatids. I usually see them in an X shape, and they're held together at a point called the centromere, which I've shown here by this gray dot. Right, in this picture, we have the human chromosomes under electron microscope. So this is what they actually look like. And you can see here sister chromatids. So on the left-hand side, we have the original chromosome, and on the right-hand side, we would have its copy. Right, now, every organism has a certain number of chromosomes in each of its cells. I'd like you to pause the video and see if you can guess which of these organisms has the most number of chromosomes. If you said humans, I am sorry, you are wrong. It is the little goldfish on the left-hand side. Right, just because you have a lot of chromosomes doesn't make you more complex. It has to do with the genes that are required to actually make that organism. Right, now notice the human has 46 chromosomes in its cells. Right, believe it or not, at one point in your life you were a single cell made up with the 46 chromosomes in the nucleus. Now those 46 chromosomes came from the chromosomes that you got from your mom and from your dad. Okay, so it makes two sets of 23 chromosomes. And now one of those sets of 23 you got from your mother's egg cell, and the other set of 23 you got from your father's sperm cell. Right, so together the two sets of 23 combined to make the 46 in the one cell that became you. Right, now a cell with both of those sets of chromosomes is called diploid, or 2N. If the cell only has one set of chromosomes, or half the number of the diploid, it is called haploid, or N. So let's say we had an organism with 10 chromosomes in a diploid cell. I want to know how many would be in a haploid cell of that organism. We know that diploid is represented by 2N, and we know that in the organism it has 10 chromosomes, right, as it's in its diploid cells. So what I want to find out is how many would be in a haploid cell, which would be a sex cell. Right, and we know that haploid is represented by N. So this becomes like an algebra problem. We're going to isolate N by dividing each side by 2. We're going to cancel out the 2's on each side, so do some division, and we get a 5 on each side. So that means in this organism's haploid or sex cells, they only have 5 chromosomes. Now we can represent all of the chromosomes in an organism with something called a karyotype that's seen here. Right, now in a karyotype, we pair the chromosomes together from the set from mom and dad based on the banding patterns of genes on each chromosome. Right, so the pair of chromosomes that were paired together based on the banding pattern are called homologous chromosomes. Right, now for these homologous chromosomes, one of the chromosomes you got from your mother's egg cell, and one of those chromosomes you got from your father's sperm cell. Okay, so they have similar genes, and together they're the homologous chromosomes. I want you to look at pair number 23, which are called the sex chromosomes. These are the chromosomes that determine gender. Right, now you have two sex chromosomes. You can either have an X or you can have a Y. If you have one X and one Y, you would be a male, and if you have two X's, then you would be considered a female.